Hi everyone, today we're going to be going through AS and A-Level Biology Cambridge. We're going to be going through the content in detail so that you know how to answer the questions that will come up. We're starting off with the AS syllabus, and that has 11 topics, and the first one is cell structure, which is quite easy. You learn about microscopes and magnification, and then you go on to the actual cell structures. So it's what you learned in, if you did IGCSE biology, that's the same structure but just more labeling and more names. So we're going to start off with 1.1, which is the microscope in cell studies. And here, so 1.1 is more practical. It's not necessarily memorizing anything, it's knowing how to use these equipments, microscope, how to prepare it, how to use it, how to magnify, how to figure out the image, the actual image, and also converting millimeter to micrometer and nanometer. This is fairly simple, so let's get started. So 1.1 is basically, all it's asking us to do here is know how to prepare to look at cells through a microscope. So you just need to know how to prepare a slide, and a slide is basically what you put underneath the microscope, how to put a cell sample inside of it and how to look at it. That's it. And there's 5 simple steps. It's very easy. So you need to be able to describe it, the step-by-step method, but it's not very detail heavy. It's pretty simple. You do need to know why we add certain things. So step 1 is essentially you get a cell, a specimen. So here we use the example of onion epidermis or cheek cells, which you actually get from inside of your cheek. We used to do this in middle school. You bite a bit of the inside of the cheeks and you put it on a microscope and you can look at it. So the sample of the cell has to be very thin, and the reason why is so that light can pass through and you can look at it easily. So the specimen, the cell has to be thin so that light can pass through it, because if it was thick it would block the light, and the image through the microscope, it would just be dark and unclear. So the thin sample basically makes sure, it makes sure that you can see the structures of the cells through the microscope. So you place the cell on a clean glass slide. Usually you use forceps, which are very thin needles, tweezers almost. So you put that onto the glass of the microscope. Once that's done, you're going to add a stain. Now, stains quite important here. The questions come up a lot about the stains. What are stains? Stains, essentially, you put a drop of it. So imagine using a pipette and dropping food colouring onto the cell. What it does is it essentially increases the colour between different parts of the cell so that you can identify it through the microscope. You can see, oh, that's the nucleus, that's the cell wall, and because many of the cell structures are transparent, see-through, and it's difficult to see using a microscope without staining it. So the staining, it binds to like specific parts of the cell, so it makes it more visible than others. It makes it distinguishable. And water, obviously, you can use water or stain depending on the cell, but water is essentially used to keep the cell hydrated and obviously help light go through. So going to stains, we have 2 stains that you need to know. So we have iodine and we have methylene. So iodine solution is used for plant cells because it stains the starch to a blue and black color and it slightly darkens. And then methylene blue is used for animal cells because it stains the DNA in the nucleus, and it also turns it blue and it helps like outline the nucleus clearly. So essentially iodine is for plant cells, methylene is for animal cells. Iodine stains the starch, turns it blue-black. Methylene, it stains the DNA in the nucleus and it turns blue. So you have to remember here that essentially the stains are to increase contrast. That's the key word, increase contrast. So after we've put the stain or the water, you're going to lower a cover slip over the specimen, over the cell that you put. So after you put the drop of stain, you're going to cover it now. So you know how the microscope So you know how the microscope glass piece, it has a bottom glass, then you have the cell, and then another piece of glass on top. That's what you're going to put it on. But again, you're going to use very, very thin tweezers, forceps. That's what they're called. They're called forceps here. And you put it on at an angle because you want to avoid air bubbles, because air bubbles are not good. So essentially you're covering the cell with another piece of glass, but you're using tweezers and you're putting it at an angle to avoid air bubbles. And then you block the excess liquid with a paper towel, just so that there's a clear view when you're looking through the microscope. We're going to go through a couple questions just to do some sort of active recall. Okay, so the first question is, why must the specimen be thin for light microscopy? So what is this question talking about? It's saying, why does the cell that we use, so a cheek cell, an onion cell, whatever cell we're going to put in to look at through the microscope, why does it have to be thin? Why does it have to be thin? It's asking us, why does it have to be thin? And this is pretty simple, because we have to let light pass through it. Light has to pass through it. And if it was thick, it would block the light and it would make the image appear dark and unclear through the microscope. It has to be thin so light can pass through. So for here, you could answer, the specimen must be thin to allow light to pass through it. This is really what they're looking for, allow light to pass through. Why do we use stains in microscopy? So we went through this earlier. Stains is like what you put on the cell before you cover it with the glass. So it's asking us, why are we using them? Why do we put it on the cell? And the key word here is to increase contrast, which essentially means you just want it to be more clear when you're looking through the microscope. You want it to have more color, more contrast. So stains are used to increase contrast, that's the key word, between different parts of the cells. So basically many cell structures, they're transparent, they are see-through, and it's very difficult to see under a light microscope without staining. So stains, it binds to specific organelles, for example, the cell wall or the nucleus. And when you're looking through, you can see that the cell wall has more contrast, so you know that that's a cell wall. So the answer here would be stains are used to increase contrast. And this really is the keyword too, increase contrast. Why is the cover slip lowered at an angle? So when we put the cell on the glass slide, add the stain, and we go to cover it with another glass slide, remember we use very thin tweezers to cover it, but we cover it when we're putting it on top of the cell, we put it at an angle. Why do we put it at an angle? Well, the reason why is if we were to just like drop it on, there could be a chance that air bubbles would be trapped underneath, and air bubbles are obviously going to like destroy it or ruin it. When you look through the microscope and there's air bubbles, it's going to like ruin it. Do you get what I mean? It's going to change the view of the cell. So we need to prevent the air bubbles, and that is why we use it. That's why we lower it at an angle, to prevent the air bubbles, because air bubbles are bad. They're going to ruin the view through the microscope. So the answer to this would be lowering the cover slip at an angle helps prevent air bubbles from being trapped under it. Prevent air bubbles is the key word here. So the next one is name 2 stains and the type of cell they're used for. We did go through this. We have 2 that you need to know. Obviously it says 2 here. So if you forgot what stains are, the question here was, why do we use stains? The stains are used to increase contrast. So the stains are used to increase contrast. And we have 2 that you need to know that I mentioned earlier, and we need to name them, and we need to name the type of cell they're used for. I did mention earlier, it's quite easy. So we have number 1, which is iodine, and that is used for plant cells because it stains starch. And then we have methylene blue, which is used for animal cells. So what you want to write here is, iodine, and it's used for plant cells, and it stains starch. So iodine is used for plant cells, stains the starch in the plant cells, and then we have methylene, and then we have methylene blue, which is used for animal cells, and it stains the DNA in the nucleus, or you could say it stains the nucleus. And this is all for better visibility. So essentially, the keywords are all of this. You need to know all of this. The name, what type of plant cell it's used for. Iodine, plant cells. Methylene blue, animal cells. It's pretty simple. So that is it for 1.1, topic 1. Subtopic 2 is draw cells from microscope slides and photomicrographs. So if you were in a school learning this, you'll have to do the actual practical work to practice this, where they'll give you a microscope and you have to look through it and look at the cell, and then you have to copy that, draw that exact cell you saw onto paper and label it. I'm not sure if they will bring an actual microscope into the exam and you have to look through it and then draw it. Maybe, who knows. But so this is for paper 3, which is advanced practical skills. This is you'll basically be asked to draw what you see. It's very similar to what you did in IGCSE biology. If you did that in paper 6, you will be given a picture and you have to copy that, maybe larger, smaller, it depends. Do you get what I mean? So you have to draw what you see, and then you have to label the cell structures accurately, and then you have to maybe possibly include like scale bars and magnification if it's known. So the common instructions here would be: make a large labeled drawing of one cell, draw and label the structure using a sharp pencil, make a clear drawing. So it's very similar to paper 6 if you did that. So what is the goal of the task? What is it testing us? It's testing you to see how well you can observe a cell and produce a biologically accurate drawing and follow proper scientific drawing conventions. So it's more than just copying what you can see. It's about accurate interpretation and proper scientific labeling. So it's pretty simple. It's a bit harder than what you had to do GCSE level, but it's not impossible. So step 1, when you see this question, you have to observe carefully. You have to look at the cell shapes. You have to look at the cell shapes. Are they like rectangular like plant cells, or are they irregular like animal cells? You have to look at the visible organelles. Can you see a nucleus? Can you see a cell wall, a vacuole? You have to look at the cell boundaries. You have to make sure you're not accidentally including two parts of two overlapping cells. And you need to make sure, are there two complete cells you can see completely as per the question. So once you've observed it, then you move on to step 2, which you have to start drawing, but there are rules to drawing. So obviously you have to have a very sharp pencil. I will link some very good visual videos on how to draw it. If you guys want me to make a visual video, I'll make that for you. I'll show you guys how to draw it, let me know. But I'm going to show you, I'm just going to tell you what you're supposed to do, but to actually see it, I highly recommend watching videos. If you're more of a visual learner, you can practice by watching videos and practice yourself by drawing yourself. It's very important to practice yourself. So let's go back to the rules. You have to use a sharp pencil. Obviously no pens, no highlighters. So this is for people that, I don't know, might have not done GCSE biology, or like they genuinely don't know. I'm going to go over the basic rules. So obviously sharp pencil only. You can't use a pen. You cannot use highlighters. You have to use clear single lines. Absolutely no sketching, shading or colouring. It has to be large and it has to be neat. It has to take up about half of the page. That's important. It has to take up about half of the page for 1 to 2 cells. And the proportions the structures must be accurately sized compared to each other. And you have to only draw what's visible. You don't invent mitochondria or chloroplasts if you can't see them, only what's visible. And you use clean, ruled label lines, horizontal, no arrowheads, you do not cross lines. So after that, you start adding labels. Depending on what you see and depending on what the questions ask, you might add labels like the cell wall, cell membrane, nucleus, cytoplasm. You do not over-label. You only label what you can confidently see, what you can confidently identify. So after you've labeled, you might have to add a title and magnification. So for example, if you have to draw an onion cell, you will write something like drawing of onion epidermal cell. And if the question says magnification is not giving, you do not give, or you do not guess. You will only write magnification not known. But if it is given, then you will write, for example, drawing of cheek cells in brackets, magnification times 400, 4000, whatever. So you add the title. I'll give you an example of a correct answer. So you'll draw two full rectangular onion cells next to each other. Each cell has a clear outline for the cell wall. Inside there's a nucleus. A nucleus is drawn as a round or oval shape. The cytoplasm is shown, but it's not shaded. And you label cell wall, nucleus, and cytoplasm with straight lines. And you write a title, and for example, the magnification is not given, so you state that the magnification is unknown. And you'd likely get 4 marks, 4 to 6 for that. You will lose marks if you sketch or shade, if you use a pen or color, if you add things that aren't visible, like guessing the mitochondria, if you don't follow the instructions. For example, drawing more than 2 cells or only partial cells, and if you use messy, unclear label lines. So that's pretty much it. It's all about practice when it comes to this, and knowing these rules and not overdoing it, only doing what they tell you to do, but in a proper way. You have to really practice drawing from real photomicrographs, watch a lot of videos on these. You have to use a ruler for your label lines, and keep your pencil sharp and do not rush. It has to be accurate and clean, not fast. So that's it for this section. Okay guys, so number 3 is telling us to calculate magnifications of images and actual sizes of specimens from drawings. So this part is very easy, very simple. It's a formula you have to memorize and conversions you have to memorize. So you would have learned this triangle thing in GCSE biology if you took it. If you didn't, you need to know the magnification formula. If you need to, because you could get asked to find either the magnification, the image size, or the actual size. And it doesn't stop there. Once you've got it, you might have to convert it from millimeters to nanometers or micrometers. But this is pretty simple once you've got the idea. So let me explain the formula if you don't know it. So essentially, what's on top is getting divided by what's on bottom. So whatever it is you want to figure out, let's say it's the actual size. Have the image size and the magnification. Then what you do, using your finger, you cover the actual size, and there you've got the formula. So can you put your finger on the screen and cover the actual size, and you've got the formula. So you put the image size over the magnification, you divide it, and you will get your actual size. Same goes with magnification. Let's say you're trying to find the magnification. Cover M with your finger, and you've got the formula. Image size over actual size. Let's say you're trying to find the image size. Cover it with your finger, you've got actual size and magnification, and when it's at the bottom, you multiply it by each other. So it's fairly simple. I would suggest memorizing this, and this is what I did. What I did in my exam is, as soon as I sat for my paper, I wrote the triangle on the pages. Then you have to know how to convert from millimeters to, this is micrometers. This symbol here, you can tell. So this is micrometers, and then it goes to nanometers. But this is very simple. I used to really struggle with this, but I made it very simple for myself as someone that's really bad with maths. But essentially, write it out like this in order from largest to smallest. Millimeters is the biggest, and nanometers is the smallest, and in the middle you have micrometers with this weird upside down H symbol. So write it out like that. Okay, and then to go smaller, you times by 1,000, and to go larger, you divide by 1,000. So wherever you are, you need to remember that if I want to go smaller, if I want to convert it smaller, I'm going to times by 1,000. If I want to go bigger, I'm going to divide by 1,000. Let's say they give you out of nowhere 50 micrometers, and they say convert it to millimeters now. So we're in the middle, we're trying to go to micrometers, which is bigger, it's larger. So what we're going to do, we're going to divide it. We're going to divide it by 1,000. 50 divided by 1,000 is 0.05. Another example, we're at millimeters. They give us 50 millimeters, and they say convert it to nanometers, which is the smallest. So we know we're going smaller, so simply we're going to times it by 1,000. So what was the number? 50 times 1,000? 50,000. So this is the order that helped me the most. I've always struggled with conversion, but I put it out like this, and this helped me. And the same with the formula. I find that these formulas are the most simple, and you do have to know this. You do have to memorize this. So we have a question here. And it says a chloroplast measures 30 millimeters. So we have the measurement, millimeters, on a photomicrograph. The magnification is 6,000. What is the actual length in micrometers? To solve these questions, what you do is you get a pen and you underline the data you have. So you underline the numbers. We have 30 and we have 6,000. And then we look. So it says a chloroplast measures 30 millimeters on a photomicrograph. What's that saying? That's the image size, not the actual size. Remember what we have. We have actual image, we have image size, and we have magnification. I didn't spell that right. So what did they give us here? They gave us the image size. It's not the actual image because it's through a microscope. This is a picture that was taken through the microscope, so it's not the actual image. It's the image size. And so it's telling us that it is 30 millimeters, and then it tells us the magnification is 6,000. So we have the magnification, which is 6,000. But then here it says what is the actual length. So we're looking for the actual length, but in micrometers. So this is what we're looking for. Let's go back to the formula. So what do we have? We have the image size and magnification. So what are we looking for? The actual size. So we're going to cover it with our finger. The formula is image size over magnification. Image size divided by magnification. Let's go back and do that. So it's image size. So we're going to do image size over magnification. 30 divided by 6,000. So let's do that. 30 divided by 6,000. 0.005. So the answer is 0.005. However, this is in millimeters. They're asking for micrometers. How do we convert? So before we go back and look, let's just think from our memory. Do you remember how to do it? We're going from millimeters, which is largest, to micrometers, which is the middle. Remember the one with the weird upside down H symbol. So what are we going to do? Okay, remember, to go larger we divide by 1,000, and to go smaller we times by 1,000. So we're trying to go from millimeters to micrometers, so it's to go smaller, so we're going to times this by 1,000, and that gives us 5. So the answer to this question is 5 micrometers. You don't write it like that, obviously you use the upside down H weird sign. So that's it for this topic. This is fairly easy. Just remember the formulas and how to convert, and that's it for this. Okay, so for 4, it's telling us to use an eyepiece graticule and a stage micrometer scale to make measurements. And basically, the big idea is we want to measure how big cells are under a microscope. With the ruler inside the microscope, which is called the eyepiece graticule, it does not have any real units on it. So it's basically like a ruler with no numbers on it, just lines. So we need to calibrate it. What does calibrate mean? It means to find out what one unit on your measuring tool actually represents in real life. So the eyepiece graticule, it goes inside the eye lens, and the stage micrometer scale, it goes on the microscope stage where the glass is. So because the eyepiece graticule, it doesn't come with real measurements, it's just lines on a ruler, you have to compare it with something that does have real measurements, and that is the stage micrometer scale. So it's like using a ruler that has no numbers on it, just marks, and you're comparing it to a real ruler to figure out, you must calibrate it. And to calibrate it, you use the stage micrometer, which is a slide with a known scale on it. It has units on it. And you compare this to this by putting them at the same, putting the zero, putting the beginning together, and you basically measure it using the stage micrometer. So how are we going to calibrate? So what do we need? Obviously we need the eyepiece graticule, which is placed in the eyepiece. We need the stage micrometer scale, which is a microscope slide with a known scale. We need obviously the microscope that we're going to look through, and then the specimen slide, which is what we looked at earlier, the cell that we made, the cell that we're looking at. So step one, we're going to place the eyepiece graticule into the eyepiece lens of the microscope. So you know where you look through the microscope, where you put your eye in, that's where you're going to put the eyepiece graticule. And the same for the stage micrometer slide, you're going to put that on the stage where the specimen slide is. So you put both of them in their places. Step two, you look through the eyepiece. When you look through the eyepiece, you'll see two rulers. You'll see the eyepiece graticule, which is just numbered lines, and then you'll see the stage micrometer, which is real scales, which is a real scale with real units. So you'll see two. Then step three, what you're going to do is you're going to line them up. You're going to basically adjust the microscopes and focus it so that both of the scales, they're visible simultaneously. And then finally, you're going to match the zero on both scales, and then you're going to see how many, you're going to figure out the unit. So you match it at zero, and then you figure out what you need to figure out. So this can come in this comes in paper 3 and paper 1 and paper 2. So it can come as a multiple choice question, it can come as a short question, but it comes a lot in practical questions. So this is again another practice one, but once you learn how to do it, it's fairly simple. There's not much to memorize. Again, it's the same as the first one we learned. You just have to learn how to do it, know what you need to do, and that's pretty much it. Okay, so number 5, which is the final topic for 1.1. This is also fairly simple once you get the idea. Here it's telling us to basically define, which means we need to know the definition. So a question could come up like, define this, you have to give the definition. So we need to define resolution and magnification. So we need to know the definition of the word resolution and magnification. And then we need to know the differences between them two. And then we're going to learn about electron microscopy, which is very simple. So what is magnification? We already learned a little bit about this. So let's say you have a tiny picture of a tiny bug. Magnification means you're making the picture bigger so you can see it better. So magnification, the definition for it would be how many times larger an image is compared to the actual object. So these definitions you do have to memorize. You can look at the mark schemes for the proper definitions. These come up often. You never know which one's going to come up, so you have to memorize them. So magnification is how many times larger an image is compared to the actual object. Magnifying, we all know what magnifying is, okay? So, but obviously making the picture bigger doesn't always mean you can see more details. It's like zooming in on a blurry picture. The bigger it gets, the more like fuzzy and like blurry it gets, right? So this is where we come with resolution. I'm pretty sure a lot of you guys have already heard the resolution, like HD resolution, it just means better quality. So, but the proper definition would be the ability to distinguish between two separate points as 2 points. But we know what it actually means. It's like how clear and sharp the picture is, right? So but what they mean here is about being able to see two tiny things close together as two things, not one blurry blob. So good resolution means a clear picture where you can see lots of little details. So we have the definition of magnification and resolution. You have to memorize them along with all the other terms that are going to come up. But it's fairly simple. We need to look at the differences between magnification and resolution. It's pretty simple. Magnification, it makes things look bigger. Resolution, it makes things look clear and sharp, right? So magnification essentially enlarges the image, whereas resolution distinguishes the fine details. You can magnify a blurry image, but resolution makes it sharp in details, right? So it's very simple. And now we're going to go into a new term, which is electron microscopes. You did not study about this in previous in IGCSE biology. Not that I'm aware of. This isn't mentioned, so it's a new topic and it's at the beginning. But it's pretty simple. So we have light microscopes. You know a light microscope, you look into it with your eye, right? You put your eye in the glass, like to look for tiny things, right? We all know that. That's what we were studying before. But now we're going to look at electron microscopes. Essentially what it is a super powerful microscope. You do not look into it with your eyes like the light microscope, but instead it sends tiny particles called electrons, which you study in chemistry. If you're doing chemistry, you'll know what they are. It sends them to look at tiny things, and it takes pictures of those tiny things using electrons, and then it shows that picture on a computer screen. You don't look in an eyepiece because electrons don't travel through air well, and the microscope needs to be inside a special place with no air, like no vacuum. So you can't just look through it with ear. You cannot just look through it like a regular microscope. You have to look at it through a computer screen. But what it does is the machine can make things look millions of times bigger and much clearer than a light microscope. So you can see tiny parts inside cells that you could not see with a normal microscope. Let's recap. Essentially, a light microscope, you look through it with your eye. You look through an eyepiece using your eye. It can make things about 1,500 times bigger. You can use it to see big parts of the cells, right? However, an electron microscope, you do not look through the eyepiece. It shows the image on a computer screen, and it uses electrons, so you can see much smaller things, and it can make things look 500,000 times bigger or more. So one is way more powerful than the other. So this is it for topic 1.1. This was mainly practical things. When you go on to 1.2, which is the last part of chapter 1, it's more about labeling the cell. So it's more of actually memorizing, and whereas this is more practical work. Make sure to practice questions on these. I hope this was helpful, and we'll continue to the next chapter.